Can you hear my voice?
Yes, madam.
Yes.
All right. Okay, so
Yes.
thank you. All right, thank you. Okay, so basically for the endomembrane system, basically it involves organelles and also structures within the cell that where their membranes are continuous. Okay, are connected to one another either continuously or discontinuously. Okay, so this it gives in this slide it shows you the properties of the endomembrane system, carry out a variety of tasks in the cell during including protein synthesis, transport of protein into membranes and other organelles, metabolism and movement of lipids, detoxification of poisons. Membrane of this system are related either through direct physical continuity by the transfer of vesicles, direct physical continuity or by the transfer of vesicles. So I have explained this slide last week. Okay, so where point number 2 and number 3 can metabolism of lipids and detoxification, it is for smooth ER. Okay, protein synthesis is for the rough ER. And transport of protein also involve the rough ER, the vesicles, the Golgi apparatus. Okay, and then the component of the endomembrane system, the membranes can involve. They are either directly connected, can direct physical continuity, or through the transfer of vesicles. Okay, so let's look at first for the endoplasmic reticulum. Okay, so it is the most extensive. Okay, extensive network of membranes account for more than half of the total membrane in many eukaryotic cells, consists of membranous tubule and flattened sac called cisternae. So tubules is for smooth ER, flattened sac, okay, is for rough endoplasmic reticulum. Okay, so the structure can the one that is folded or in the tubule form, we call it as cisternae. Okay, so it has space inside the cisternae, we call it as cisternal space. Okay, which is separate the cisternal space will be separated from the cytoplasm by the ER membrane. Okay, so the ER membrane, it is a single layer. It is continuous with the outer membrane of nuclear membrane. So if you look at the structure, you can see that the membrane of the rough ER is continuous. It is continuous with the membrane of the with the outer membrane of the nuclear envelope. Outer membrane only. Okay, so nuclear envelope, it has double layer membrane. So the outer membrane is directly connected to the membrane of the rough ER. Membrane of the rough ER is directly connected to the membrane of smooth ER. Okay. So next, okay. So two types of endoplasmic reticulum. We have the smooth ER, okay, which doesn't have any ribosome, okay, and then the rough endoplasmic reticulum, it has a ribosome, okay, which is bound to the membrane, facing the outer surface, facing the cytoplasmic side, okay. So I've drawn this already during the lecture. And so when you draw, when for example you are drawing the rough endoplasmic reticulum. So this is, for example, one portion, a portion of a rough endoplasmic reticulum. So the drawing of the ribosome, which is bound to the membrane, must face outside. It must face outside, facing the cytoplasm. Okay, not inside in the lumen. Okay, so itu salah lah, kan? Salah. Okay, so and then on the smooth and endoplasmic reticulum doesn't have any ribosome. It is interconnected tubules. As you can see, the structure is it is tube-like, kan? Interconnected tubules, and then it is continuous. The membrane of the smooth ER is continuous from the rough ER, and it doesn't involve in the process of protein synthesis. Why? Because it doesn't have any ribosome. Okay, it doesn't have any ribosome. Um, okay, so next, okay, next one, we are going to look at the function of the smooth ER. Okay, so function of smooth ER, mostly as you can see from the list, it involves in the metabolism of lipids, can the synthesis of lipids. Okay, so metabolize lipid, synthesize cholesterol, as we know it is also lipid, and then also synthesizes phospholipid. Okay, and then because it synthesizes cholesterol, so therefore it also involved in the synthesis of steroid hormones such as testosterone. Okay, and then it also involves in the absorption, synthesis, and transport of fats, detoxify toxin, okay, and then also breakdown of glycogen from glucose, okay, in the liver. Can so in the liver it will have many smooth ER to break down glycogen to form glucose. Okay, so that is on the smooth ER. So now we are going to look at the rough endoplasmic reticulum. So rough endoplasmic reticulum, the membrane is flattened sac. Okay, so also known as cisternae. So protein from bound ribosome. Okay, so the membrane of the rough ER. So this is the rough ER. Can the rough ER. So it has ribosome embedded. Okay, embedded in the membrane of the rough ER, facing the cytoplasm. So the function of the ribosome is to synthesize protein into the lumen of the rough ER. Okay, so ribosome, which is bound to the membrane of the rough ER, function to synthesize protein, and the protein will end up in the cisternal space. Okay, so the protein produced will also get attached with carbohydrates to form glycoproteins. Okay, glycoprotein is the carbohydrate parts. Protein are synthesized from the ribosome. So glycoproteins, right? So glycoprotein is an example of secretory protein. Secretory protein are proteins to be secreted out of the cell. Okay, so and then is then okay, it then departs or buds off from the ER in a membranous vesicle called as transport vesicle. As you can see from this drawing, and so this is the rough ER. So the ribosome synthesizes the protein, and then the protein will be packaged into vesicle. Okay, into vesicles. So the vesicle we call it as transport vesicles. These are transport vesicles. So the function of the transport vesicles is to transport the product of the rough ER. Product is referring to the protein or the glycoprotein. Okay, so transport product of rough ER, proteins or glycoprotein, towards the Golgi apparatus. So the function of the Golgi apparatus is to receive the transport vesicles and then to modify the protein inside. Okay. So this transport vesicle transit from one part of the cell to another. From one part of the cell to another means that the transport vesicles originates, or it comes from the rough ER and then goes to the Golgi apparatus, fused with the membrane of Golgi apparatus, and then the product will end up in the Golgi apparatus to be modified and then packaged again to form another transport vesicle, or we can also call it as secretory vesicle. Okay, secretory vesicle that will go to the plasma membrane, fuse with the plasma membrane to secrete out the product. So the product can be hormones, it can be enzyme, or it can be other proteins that the cell produces. Okay. So and then another function of the rough ER, it acts as membrane factory for the cell. Okay, it acts as a membrane factory for the cell. So by adding membrane protein and also phospholipid to its own membrane in making secretory protein. Okay, we know that the ribosome, the ribosome which is bound to the membrane of the rough ER, synthesizes protein. Okay, it synthesizes the protein. So the protein will get modified in the Golgi apparatus. So this protein can either be embedded in the membrane, okay, in the membrane of the Golgi apparatus, and this region will form transport vesicle, okay, transport vesicle or secretory vesicle. So when this transport vesicle or secretory vesicle fuses with the membrane of the plasma membrane, so the protein here will become part of the protein embedded in the plasma membrane of that cell. So this protein it has several functions. It can function as, for example, identification tag for the cell, antigen for this. Antigen for the cell. Okay. Uh, so So identification tag it can function as transport protein, transport protein that are embedded in the membrane. So later in chapter 3, we are going to look at various types of proteins that are embedded in the plasma membrane, and this protein is produced by this mechanism. Okay, initially it is synthesized by the ribosome in the RAF-AR, transport to via the transport vesicles, modified in the Golgi apparatus, and then packaged again into vesicle and becomes protein that are embedded in the plasma membrane, or it can become protein to be secreted out as hormone or enzymes. Macam tu, okay? So it depends. Or the protein can become enzyme in the lysozyme, which function within cells. Okay, so it depends. It has many different types of function. It produces protein that has different types of function. Okay, so it becomes the protein can become lysozyme in lysosome. It can become hormone or enzyme to be secreted out. It can become a protein that are embedded in the plasma membrane that has several different functions that we are going to look at in chapter 3. Okay, so and then another thing for rough endoplasmic reticulum tadi balik semula kepada point ni, is that it function as membrane factory. Why is that? Because as you can see throughout the process yang saya explain tadi, saya padam baliklah semua ni. Okay, kan? So as you can see, the membrane of the rough endoplasmic reticulum, okay, involved in the formation of membrane for the transport vesicle. Okay, so the membrane of the transport vesicle will then fuses with the membrane of Golgi apparatus, becomes the membrane of the Golgi apparatus. Okay, the membrane of the Golgi apparatus then forms the membrane of secretory vesicle, another transport protein, which fuses with the membrane of plasma membrane. So that's why here, it says here, rough endoplasmic reticulum will function as membrane factory for the cell. Daripada membrane RAF-AR lah dia akan hasilkan membrane transport vesicle, dia akan jadi part of membrane of Golgi, dia akan jadi part of the membrane of the lysosome, it becomes part of the membrane of the plasma membrane also. Kan? So that is the explanation for the last point here, membrane factory for the cell. Adding membrane protein and phospholipid to its own membrane in making secretory protein. We are going to look at also, kan, so the product of the Golgi apparatus that produces transport vesicles containing protein. So these transport vesicles can also migrate back to the RAF-AR and fuses with the membrane of RAF-AR, becomes also part of the membrane of the RAF-AR. So dia macam tu lah, kan, so dia boleh patah balik vesicles, dia boleh patah balik ke rough endoplasmic reticulum, or dia boleh patah balik juga becomes part of the membrane of the Golgi apparatus. Okay. Okay, so semua faham ke yang point last ni? Ada yang tak faham? Okay. For this okay.
Kurang faham Madam.
Kurang faham. Okay, dia macam ni. Rough endoplasmic reticulum, kan, dekat membrane of rough endoplasmic reticulum ada apa? Ada ribosom, kan. Bila dia ada ribosom, dia akan hasilkan protein. So yang merah ni protein, yang warna pink wavy light ni protein juga. Yang warna hijau ni can also be protein. So this protein can be whole protein, it can be and it can also become glycoprotein kalau dia bergabung dengan karbohidrat. Okay. So produk semua ni nak pergi mana? Dia nak pergi mana? Dia nak pergi kepada next stage, which is in the Golgi apparatus. Untuk produk ni sampai ke Golgi apparatus, mesti dia dipackkan dalam bentuk vesicle. To form this vesicle, kan? So the membrane of the vesicle will be synthesized from the membrane of the rough endoplasmic reticulum. Hari tu saya dah draw dah, kan? Sekejap. Nampak ni tulisan kan? Yang ni. So the membrane of the RAF-AR that has ribosome attached, it will bud off. Kita guna perkataan bud off. Okay, bud off or departs, okay, to become membrane of the rough ER, and then membrane of the transport vesicles, and then this transport vesicle, its membrane will fuse with the membrane of the Golgi apparatus. Dia akan fuse. Dia adalah sebahagian daripada membrane Golgi pula. Lepas tu nanti produk akan masuk ke dalam Golgi apparatus to be modified and then packaged again into another transport vesicle. As you can see, in order to produce this transport vesicle, the membrane comes from the membrane of the Golgi, which initially comes from the membrane of the rough ER. Okay, so this transport vesicle that contains the product, okay, the modified product, which is protein, now fuses with the membrane of plasma membrane, becomes part of the membrane of plasma membrane. So that's why the rough endoplasmic reticulum is the membrane factory for the whole cell. Macam tu. Boleh faham
Faham.
Okay. Kat situ. Okay, so on the Golgi apparatus or the Golgi body, so you can use both terms. So for the Golgi apparatus, function as receiving and shipping center. Receives what? Receives transport vesicle coming from the rough ER. Okay, and then as transport vesicle, the membrane fuses with the Golgi apparatus membrane. Protein will enter into the Golgi membrane to be modified. So kat sini boleh tambah kat sini. Modify the product of rough ER and then ships the protein or the product into another vesicle. So that's why receiving and shipping center. Okay. So consists of flattened membranous sac called as cisternae. Sama juga macam rough ER tadi ataupun smooth ER. So the structure kan structure yang berlipat-lipat tu, so we call it as cisternae. Okay. So each Golgi stack has three areas, has three regions. Cis phase kan is the entry phase. This one it faces the rough endoplasmic reticulum. Okay. And then the medial region is the middle region here, where modification occurs. And then transphase is the exit phase. So this faces the plasma membrane. The transphase will produce secretory vesicle or another transport vesicle, and this vesicle will go towards the plasma membrane and fuses with the plasma membrane to release the products out of the cell. Okay, the product here is referring to the protein lah, the modified protein kan. So sama ada yang dia akan jadi hormon ke, dia akan jadi enzyme ke, whatever product that originally comes from the rough ER. Okay, and then function of the Golgi apparatus is to modify products of ER. Products of rough ER is either protein or glycoprotein lah kan, glycoprotein to be modified in the Golgi apparatus kan. So manufacture certain macromolecules kan, and then certain macromolecules is the glycoprotein lah. So from the glycoprotein it may becomes enzyme, it may becomes hormone, it may become lysozyme in the lysosome, or it can become transport protein to be embedded in the plasma membrane. Any proteins that are embedded in the plasma membrane that have several functions. Okay. So and then the product protein, glycoprotein, which is made in the rough endoplasmic reticulum, okay, will be modified, manufactured and sorted, packaged, okay, and then packaged into another transport vesicle kan, another transport vesicle. So the Golgi apparatus, it produces transport vesicles that has the modified product, modified protein. Okay, so apart from it becomes transport vesicle, it can also become lysosome. Okay, it can also become lysosome. The vesicle can also be called apart from transport vesicle, dia boleh juga kita label sebagai secretory vesicle. Okay, secretory vesicle or transport vesicles. So boleh slash kat sini, secretory vesicle, and it can also become lysosome. Okay. Okay, so if you look at this diagram here, it shows you the structure of the Golgi apparatus. Okay, so for the Golgi apparatus, you have to take note that Golgi apparatus has three regions. The cis phase facing the ER, rough endoplasmic reticulum kat atas ni. Kat sini lah ada nukleus kat atas ni, ada rough ER kan. So the cis phase, it faces the rough endoplasmic reticulum ready to receive any transport vesicles coming from the rough ER. Okay. And then it has medial region for modification to occur, modification of protein or the product of ER. And then the final product would then again be packaged into another vesicles. Ni yang saya mention tadi. So these vesicles can be transport vesicles. You can also call it as secretory vesicles, or it can either become lysosome. Yeah, lysosome. Okay. So those are the type of vesicles produced or coming out of the Golgi apparatus. Kan? As you can see, this membrane of the vesicle comes from the membrane of the Golgi apparatus. Macam tu, okay? So, as you can see, okay, so another region kat sini is transphase. So transphase, it faces the plasma membrane. Kan? It faces the plasma membrane ready to accept or fuse with the transport vesicles. So if I were to draw this clearly, kan? So this is the transport vesicle or secretory vesicle. And then it goes to the plasma membrane, fuses, okay, fuses with the plasma membrane and releases the product out of the cell. So this product can be enzyme, it can be hormones that are made up of proteins such as insulin, or it can become proteins that are embedded in the plasma membrane that function as maybe transport protein, can to transport substance in and out of the cell, okay? Or it can become enzymes that are also embedded in the membrane. So the function of this protein embedded in the plasma membrane, we are going to look at in chapter 3, okay? So this is the whole process. I've explained this. So let's look at point by point lah. So the first one, vesicle moves from ER to the Golgi apparatus. So this is transport vesicles which contains products. Product here is referring to protein or glycoprotein it comes from the rough endoplasmic reticulum. Okay, so this is the transport vesicles. And then these transport vesicles, kan, you all letak kat sini nak bagi jelas transport vesicle lah, kan. Transport vesicle, another word you can replace here for you to easily understand, instead of coalesce, you can use the term fuse, kan. Fuses, okay. The vesicles, the membrane of the vesicle fuses with the membrane of the Golgi apparatus at the cis phase. Okay, cis phase. And then it says here cisternal maturation. Golgi cisternae moves in a cis to trans direction. Okay, so the movement of the product, again, which is end up in the lumen of the Golgi apparatus, will move from cis medial region to trans phase. So as you can see, the membrane of the transport vesicle fuses with the membrane of the Golgi apparatus. The product will end up in the lumen of the Golgi to be modified, okay, and then packaged again at the trans phase. Okay, at the trans phase here. Trans phase, this is trans phase, another trans phase facing the plasma membrane, okay. So, and then vesicles form and leave the Golgi carrying specific product to other locations or to the plasma membrane for secretions. It says here other locations. Other location is within the cell. So this vesicle, as I mentioned before, this vesicle will either go to the plasma membrane to fuse, or it becomes lysosome. Okay, so we can assume this is lysosome, or it can the vesicle can go up towards the cis region of the membrane, fuses on the upper part of the Golgi, become parts of the membrane for the Golgi apparatus also, or it can even go to the rough ER, fuses with the membrane of the rough ER, become parts of the membrane of the rough ER. So it depends on the function of that vesicle. Okay. So so summary for the secretory protein transport. So it says here polypeptide synthesized on ribosome bound to the rough ER. So this is rough ER. Saya padam balik. I'm going to erase out all this. Okay, to go through all the process. Okay. Polypeptide, okay, synthesized on the ribosome of the rough endoplasmic reticulum. As you can see from the structure, it has many ribosome bound, okay, to the membrane of the rough ER. The function of the ribosome is to synthesize protein into the lumen of the rough ER. We call it as cisternal space. Okay, so and then assembled and joined with carbohydrate to form glycoprotein. Okay, glycoprotein. And then the terms here glycoprotein, and then package at the transitional region. So this is what we call it as transitional region. Sekejap kelas. Sekejap kelas. Okay, sorry class. Okay, back to point 3. Okay, so back to point 3. Okay, so protein produced from the ribosome, okay, can either join with carbohydrate to form glycoprotein, and this glycoprotein will be packaged into vesicle at the region of rough ER. We call it as transitional region. Kawasan rough ER yang akan terhasilnya transport vesicle, kita namakan dia sebagai transitional region. Okay, transitional region. From there, it contains glycoprotein, and this protein will be packaged into transport vesicles that will migrate to the Golgi apparatus. Okay. So transport vesicle move protein to the Golgi apparatus towards the cis phase of the Golgi apparatus, and then the protein will be modified in the Golgi apparatus from cis to trans phase. Okay. And then proteins such as glycoprotein or secretory protein. Secretory protein here can be enzyme. It can be hormones. Hormones that are made up of proteins such as insulin. Ada hormon yang komponen dia protein. Ada hormon yang komponen dia adalah lipid. Okay. So secretory protein are packaged in transport vesicle at the trans phase. So kalau ER tadi transitional region. Kalau Golgi, we call it as trans phase. Okay. So transport vesicles that departs from the trans phase contain secreted products inside. Okay. So from the vesicles that bud off at the trans phase, at the trans phase here, will contain products. Okay, so products. So the vesicle may fuse with the plasma membrane and release the content from the cell. So this content can be inilah enzyme ke, hormone ke, or whatever protein that the cell produces, which are proteins. Okay, and then it also can give rise to specialized other organelles such as lysosome and also vacuoles. Kan? So kalau dalam plant, contohnya kita ada central vacuole. So this is how the central vacuole are made. Or kalau kita ada dalam contohnya protists, we have contractile vacuole. So this is how the vacuole are made. Okay. And then, or it can also produce vesicles that fuse with less mature Golgi cisternae, like the previous slide here, as you can see. So the vesicle can go up to the cis phase of the Golgi apparatus, fuses with the cis phase and become parts of the membrane at the cis phase. Okay, and then also carry protein back to the ER required in the ER. Kalau you all tengok kat sini, any membrane inside cell, especially the one that involves in the endomembrane system, they will have proteins embedded. Macam contohnya kat sini kan, protein embedded. As you can see, protein embedded in the membrane. Kan? So these protein that are embedded in the membrane has functions. Kan? So one of the function of this protein embedded, it function as transport protein. Transport protein. One of the function of transport protein is to transport ion across membrane. Kan ion across membrane akan ada ion keluar masuk ke contohnya ion keluar masuk or other molecules that are polar. So in order to allow the transportation of this molecule, it has to use transport protein. So kita pause kejap dulu kelas. Okay. So let's okay. Those are the basic components of the endomembrane system. Okay, so from the components of the endomembrane system, from the vesicles that produces at the transphase of the Golgi apparatus, the vesicle can become lysosome. Yeah, so it can become lysosome, it can become vesicles, it can become transport vesicles. Can so in this case, the vesicle that bite off from the transphase of the Golgi apparatus may become lysosome. Okay, so lysosome is a membranous sac of hydrolytic enzyme that can digest macromolecules. It has hydrolytic enzyme, so we call it as lysozyme. Okay, so lysosomal enzyme can hydrolyze protein, fats, polysaccharides, and also nucleic acids. So the enzyme of the lysosome will work best in acidic condition. Okay, usually at pH 5. Okay, so if the lysosome breaks, the release enzyme are not effective as the cytosol S has a neutral pH. Okay, if a lysosome break, if one or a few lysosome break, but if you have an enormous amount of lysosomal to break, it can cause the whole cell to undergo autolysis. Okay, autolysis. However, lysosomal membrane becomes fragile when the cell is injured, lack oxygen, and when an extensive amount of vitamin A present causes lysosome to rupture and leads to autolysis. So lysosomes will rupture and releases a lot of lysosomal enzyme and causes autolysis. Okay. Okay, so function of the lysosome, it carries out intracellular digestion. Intracellular digestions are digestion that occur within the cell. Okay, within the cell. So the first one here is phagocytosis. Okay, what is phagocytosis? A process by engulfing food particles, and it causes the formation of food vacuole. Okay, so food vacuole will then fuses with the lysosome that contains hydrolytic enzyme to digest the food inside the food vacuole. Okay, example of cells that undergo phagocytosis are white blood cells. Okay, white blood cells or protease. Okay, so protease will engulf the food outside, or if it's white blood cell, the pathogen can into the cell. So for example, we are focusing on white blood cell lah, kan? So white blood cells, the function of white blood cell is to engulf pathogen. Engulfing the pathogen, okay, which is considered as food right now, okay. So the process is phagocytosis. So if you look at this diagram here, so this is the membrane of the cell, membrane of the white blood cell, for example, or membrane of the protease, for example. So the food is outside, okay, the pathogen is outside of the cell. So this food has to be internalized. So to internalize the food, so the plasma membrane will fold inward. Okay, plasma membrane we see here is it fold inward. Okay, fold inward to take in the food into the cell, and then the food will be surrounded by the membrane originated from the plasma membrane. So now it become food vacuole. Yeah, so the membrane of food vacuole was from the membrane of the plasma membrane. Okay, so the food vacuole that contains the food will fuse with the lysosome. Okay, with lysosome that also has membrane. So membrane of the lysosome fuses with the membrane of the food vacuole. Enzyme within the lysosome will digest the food inside, and then the process of digestion occurs, which is intracellular digestions. Okay, so digestion will break the food into smaller food particles, into smaller molecules, and this molecule will be absorbed into the within the cell. Okay, for the usage of that cell. Okay, so and then another one
Dalam,
here,
boleh tak saya nak tanya tadi, autolysis tu macam mana?
lysis tu pecah kan, autolysis dia proses tu automatically digest the cell, destroy the cells. Boleh faham ke?
Boleh. Faham.
Sekejap Sekejap. Okay, the statement kat sini it says, yang first sekali saya Google, autolysis is the enzymatic digestion of cell by the action of its own enzyme. Okay, and it mostly occur in dying or dead cells. Okay, lysosomes are responsible for autolysis. They contain hydrolytic enzyme that digest carbohydrate, protein, lipids, foreign particles, and dead cellular materials. Kalau lysosom tu satu je break, dia tak bagi efek kepada sel. Tapi kalau macam contohnya keadaan yang macam tadi menyebabkan large amount of lysosome to break, it can lead to autolysis. Okay. Kalau kita tengok kat sini, another one, autolysis means self-destruction. Auto self-lysis rupture. Okay, sempurna. Right? Ada banyak lah explanation kat sini, alright? The process of self-destruction of a cell organelle or tissue. Macam itulah, alright? Okay, so next. Okay, so phagocytosis, kan? And then next one is autophagy. Autophagy is recycles the cell's own organic material. Damaged organelles become surrounded by membranes. Lysosome fuses with this vesicle. Lysosomal enzyme dismantle the enclosed material and releases it into the cytosol for reuse. Okay, so for autophagy, it means what? Inside our cells, inside individual cells, there will be many organelles. Okay, so this organelle at some point it will come to a point that it needs to be destroyed. Sebab dia dah lama rasa tu kan, it doesn't function anymore. So the cell needs to renew the organelle. So this old organelle, worn out organelles, can, such as for example in this case in the diagram, as you can see, there's the peroxisome or the mitochondria, kan. These are considered as worn out organelles yang nak dipusnahkan. So in order to destroy these worn out organelles, so this organelle will be wrapped around or surrounded by 2 layers of membrane. Okay, they are surrounded by 2 layers of membrane. So this form vesicles containing the worn out organelles. So in order to digest that worn out organelle or to break down the worn out organelle, so the outer membrane of the vesicle that contains the worn out organelle will fuse with the membrane of the lysosome. Enzyme within the lysosome will digest the content inside the organelles lah, kan. So into monomers, into smaller molecule that the cell can absorb and reuse. Macam tu kan, absorb and then reuse. That is autophagy. Recycles the cell's own organic material. Okay. So the organelle dah lama breakdown by fusing with the lysosome and then reuse the matter. Okay, so that is autophagy. Okay. Okay, so next one is vacuoles. Again, another component of the endomembrane system are vacuoles. So vacuoles are large single membrane enclosed sac. Any organelles of the endomembrane system, they only have single membrane. Apart from the nuclear envelope lah. So nuclear envelope hanya terlibat the outer membrane, kan. Outer membrane of nuclear envelope directly connected to the membrane of the rough ER. Okay. So anyhow, vacuoles, okay, vacuole is single enclosed membrane sac. So a plant cell or fungus cell may have one or several vacuole. So there's 3 types of vacuole. Vacuole ni dia sama lebih kurang macam vesicle. Apa beza dia adalah vesicle, the size of vesicles is smaller compared to vacuole. Vacuole, the size is bigger. Vesicle, the size is smaller. So that's why kita guna term vesicle. Vesicle smaller in size, vacuole is larger in size. Okay. So 3 types of vacuole. One is food vacuole, which are formed from phagocytosis, tadi by the white blood cells. Okay, white blood cells, by the proteins that engulf its prey, okay, makanan dia lah, kan. So and then another one is contractile vacuole, found in many freshwater protists to pump excess water out of the cell, which is in the process of osmoregulation. and then central vacuole, which is found in plant cell. Okay, so the membrane of the central vacuole, we call it as the tonoplast. So basically, kita go one by one. Okay, so this, as you can see, is the okay. So this is the organism, we call it as paramecium. Okay, so paramecium is unicellular eukaryotes. Okay, unicellular eukaryotes. Because it is eukaryote, it has membrane-bound organelle. So one of it is the contractile vacuole. Kan? So the paramecium, dia kena makan. Okay, so cara dia makan adalah through phagocytosis. And then phagocytosis leads to the formation of food vacuole. Okay, and then contractile vacuole for osmoregulation. Proteus is an organism, for example, that lives in fresh water. Okay, so because they live in fresh water, so the water from outside will have a high tendency to enter into the paramecium. So this excess water needs to be pumped out. Okay, so the pumping out of that excess water by the contractile vacuole. Okay, so and then another type of vacuole is the central vacuole, which is found in plant cell, which is to cause plant cell to be in the turgid condition. Kan, as the central vacuole is capable of storing water, kan, to cause a plant to be turgid. Okay, so the normal condition for plant to be in is turgid. So that's why you have to water your plant. Kan, you have to water the plant. The plant cannot receive less water than it is supposed to. So this excess water will be stored in the central vacuole. Central vacuole is large. It pushes all the other organelles to the peripheral side of the cell. So when you are doing experiment 2, for example, like S10, so they have observed that the nucleus, okay, the nucleus of plant cell will be found at the edge of the cell, at the peripheral side of the cell, due to the presence of large central vacuole. Okay, so okay, so this is to summarize the whole thing lah, kan. So you can go through this slide to summarize the component of the endomembrane system, starting from the outer membrane of the nucleus that is continuous with the membrane of the rough ER, that is continuous with the membrane of the smooth ER. From the membrane of the rough ER, at the transphase, it forms the transport vesicle containing protein of the rough ER, goes to the Golgi apparatus, fuses at the cis phase. The protein will be modified and then packaged again at the transphase, either fuses with the plasma membrane to secrete out all the protein products, or it can become lysosome, or it can fuses with the membrane of the rough ER, or it can even fuses with the membrane of the rough ER. Macam itulah. Okay, so this is the summary of the endomembrane system. I want you to go through again the endomembrane system, and you have to understand each component of the endomembrane system. So do you have any question regarding to this lecture? Kita stop dulu sampai sini. Nanti hari Jumaat kita sambung the rest of the chapter. Any question?
No.
Tak ada So kita stop dulu kelas. Thank you ya. Thank you all.
Thank you, madam.
Okay.
Thank you, madam.
Thank you, madam.
Thank you, madam.
Okay.
Thank you, madam.
Okay.
Madam,
Ya.
saya ada soalan.